Everybody, my name is Jonathan Tenenbaum, and today I will be presenting the research I conducted at the Skirball Institute of Biomolecular Medicine at New York University. My work is titled Targeting Nrf2 for Regulation of Chemokine Gene CCL2 and the Promotion of Diabetic Wound Healing through CHIP. To begin, what are diabetic ulcers? Diabetic ulcers are open sores or wounds that occur in approximately 15% of diabetic patients, 6% of which will be hospitalized due to diabetic ulcer-related complications such as infection. In fact, in the United States, every 17 seconds somebody is diagnosed with diabetes, and every day, 230 Americans with diabetes will suffer an amputation as a result of untreated diabetic ulcers. They are commonly located at the bottom of the foot, and as you can see in the diagram I've included below comparing normal wound healing to diabetic wound healing, in the diabetic wound, macrophages are unable to infiltrate the site of injury, preventing them from carrying out the wide host of functions that make them so useful to wound healing, such as reducing inflammation, removing dead cells, and increasing tissue proliferation. Recently, the large-scale study linking the effects of foot ulcers and diabetes was published in Diabetologia, one of the largest diabetes science journals, and this study proved that diabetic patients who develop foot ulcers are at a higher risk of dying prematurely than those who do not. Ultimately, there have been little to no treatment options provided to or offered to diabetic patients to aid diabetic wound healing that work at the biomolecular level. My research seeks to establish a basis upon which possible treatment can be developed. When I arrived at the lab, my mentor explained to me his identification of an antioxidant response element sequence within the promoter site of CCL2. CCL2 is a chemokine gene, essential to macrophage response orchestration, a function that, as I pointed out in the last slide, is characteristically defective in diabetic patients. Once CCL2 is activated, it initiates the production of CCL2 chemokines that attract white blood cells to the site of injury. When my mentor and I were discussing what could possibly bind to this ARE site, and thus activate CCL2's role in wound healing, we decided upon researching Nrf2 as a possible candidate. Nrf2 is a transcription factor that plays a crucial role in reactive oxygen-species balance and the transactivation of critical response genes. To the right, I have pictured what a possible Nrf2 ARE pathway would look like. After Nrf2 binds to the ARE site, the transcription and translation of the genes that follow the site in sequence begins, and this is terminated once KAP1 binds to and thus degrades Nrf2. Thus, the purpose of my research is to prove the existence of this hypothesized Nrf2-CCL2 relationship so that new doors can be opened for injury response initiation and/or expedition through forced activation in diabetic wounds. Below I've included a diagram demonstrating the marked decrease in percent wound size in a diabetic wound after treatment with CCL2 chemokines after 10 days. Before we can discuss the specifics of the CHIP procedure, I want to note that I was provided one sample of wounded and one sample of intact epithelial tissue in order to prove that this possible relationship between Nrf2 and CCL2 is only activated in response to the presence or occurrence of a wound. Each sample I ran independently through the CHIP procedure. Now for the nitty-gritty of CHIP. CHIP, aside from being a fun acronym, is a procedure by which a protein is determined to bind to a specific DNA sequence in vivo. This begins with cell lysis, which entails the rupturing of the cell's membranes so that the DNA can be extracted and utilized. Then, cross-linking, which my mentor performed because it requires the use of hazardous chemical formaldehyde. Cross-linking forms covalent bonds between the nucleic acids of the proteins and the DNA, forming more stable DNA-protein complexes that will remain intact in the following steps. Then I carried out shearing, which I used a sonicator to fragment the DNA with proteins actively bound to it into more workable pieces, around 500 base pairs, by applying bursts of sonic waves to the DNA. I then carried out immunoprecipitation by adding Nrf2-specific antibodies that bind to the Nrf2 proteins bound to the DNA. These antibodies are attached to dense beads that allow for the precipitation of only the DNA segments bound to by Nrf2 through centrifugation. I then reversed these crosslinks and cleaned up the DNA. I then carried out quantitative PCR, in which I used real-time PCR to determine if the known CCL2 ARE sites were specifically enriched by immunoprecipitation. Last, I want to briefly review the control network I employed to ensure result verity. I performed the same CHIP procedure for an immunoglobulin G or IgG gene proven against Nrf2 binding as a negative control. I did the same for an NQ1 ARE site known to be bound to by Nrf2 as a positive control. And finally, I did the same preparation for an NQ1 gene proven against Nrf2 binding to corroborate the negative control and serve as yet another means for comparison. This brings me to the results of my experiment. To begin, picture the results for the CHIP procedure I performed upon the sample of intact epithelial tissue. As you can see, the results for the CCL2 site immunoprecipitation in terms of enrichment over negative control IgG does not compare considerably enough to the positive control and is about equal in terms of percent enrichment over negative control to the negative control itself. Thus, we cannot say that in the intact epithelial tissue that Nrf2 binds to the ARE site of CCL2. However, in the figure comparing the results for the intact and wounded tissue, it can be seen that the results for the CCL2 ARE site compare significantly to that of the positive control performed for the intact and compares even more so in terms of percent enrichment over the positive control performed for the wounded tissue sample. Here, I'm showing you here with my mouse the difference, how these values compare, how this value for the CCL2 ARE site compares considerably to that of the positive control in the wounded and in the intact. So what conclusions can I draw based on my results and the events of my research? Several conclusions can be drawn. First, Nrf2 is proven to bind to the ARE site and thus activating CCL2 as demonstrated by the comparison of chip results of the Nrf2 CCL2 ARE site to the positive NQ01 ARE site control and enrichment over negative control IgG. However, as demonstrated by the difference in the results between the wounded and intact tissue, this relationship is only activated in response to the occurrence of wounding as demonstrated by the insufficient enrichment over IgG of the CCL2 ARE site as compared to the positive and negative controls in the intact tissue sample. Lastly, Nrf2 activates the ability of CCL2 to produce chemokines that attract white blood cells to the site of wounding in response to injury. These conclusions provide for discussion in that if the period where Nrf2 is bound to the ARE site of CCL2 can be extended, this can extend the period where CCL2 is enabling the active production of chemokines that attract white blood cells to the site of injury. This could expedite wound healing and can be especially beneficial to diabetic patients who face hospitalization or amputation as a result of their body's inability to manage macrophage response. Thus, this relationship can and should be manipulated. Finally, before I take the leap to discuss the future application of my research as a potential treatment option, I would like to conduct a study involving more samples of intact and more samples of wounded tissue in order to prove with increased certainty that this Nrf2-CCL2 relationship exists in most, if not all, cases. Furthermore, regarding my research's application as a treatment option, the eventual goal of my work, through literature research and discussion with my mentor, I began to consider the possibility of modifying proteins such as P21 and P62 to bind to Nrf2 proteins actively bound to the CCL2 ARE site. These proteins are able to temporarily prevent the binding of KAP1 to Nrf2, and thus prevent or postpone the degradation of Nrf2 and the cessation of the transcription of the genes that follow in sequence. In this case, this would postpone the cessation of the transcription and translation of CCL2. Thus, this would extend the period where CCL2 is allowing the body to actively produce chemokines that attract white blood cells to the site of injury. The reason why it's important that these proteins are able to bind to the Nrf2 that are actively bound to the ARE site of CCL2 is that Nrf2 regulates a wide host of other genes and monitors a wide host of other genes that are involved in a variety of purposes or functions. For example, I've read extensively in my research about how the Nrf2 pathway plays a role in preventing obesity and encouraging weight loss. So I intend in my future research to modify these proteins to bind specifically to the Nrf2 that's bound to the ARE site of CCL2 so that I can elongate this period where the body is producing CCL2 chemokines without affecting the wide host of other genes that Nrf2 monitors, and their regular monitoring is crucial to the activities of the body, and I would not want to affect these interactions. I would solely be able to use this relationship that I've proved for the benefit of these diabetic patients that suffer from the consequences of their body's inability to manage macrophage response. Thus, here are my references, and I want to thank you all for listening. I want to thank you also for holding this event in spite of the present circumstances. I also hope that you and your loved ones and everyone around you is happy, healthy, well, and safe. And just thank you for the opportunity despite the crazy state the world is in. All right. Thank you now. Have a nice day.